This is part 1 of our lecture for Chapter 5, and we are also including 4.3, which is on functional groups. So this is going to focus on the chemistry of carbon, or otherwise known as organic chemistry. So first of all, a reminder of why we study carbon, and because of its important role in life. While 72% of cells are made up of water, the big majority of what's left is made up of carbon compounds. And then we have a small amount, 3%, made up of salts. So organic chemistry is the study of carbon compounds. And again, a reminder, carbon atoms are versatile building blocks because of their bonding properties. Carbon can make those 4 stable covalent bonds because carbon has a valence of 4. So it has 4 electrons in its outer valence, outer electron shell, and that leaves 4 spaces or valences to make 4 nice bonds. And that symmetry of having 4 available bonds helps carbon have a lot of structural significance when it comes to the bonds that it forms and then the actual molecules that can be formed based with a carbon framework. So when we are studying biochemistry or organic chemistry, we want to start by looking at the most simple and very common bonds, which are hydrocarbons. So that would be the combination of carbon and hydrogen. And these are very plentiful in all organic biomolecules. They're very nonpolar, which means they're not going to be soluble in water, and that means they're also hydrophobic. Because of this property of being nonpolar, hydrophobic, not soluble in water, they are very stable and they have very little attraction between molecules. So most are going to be a gas at room temperature. So when I'm saying most, what I mean are molecules that have large amounts of hydrocarbons. So if you look at the examples in this box here at the bottom, you can see ethane, propane, butane, isobutane. These are all very flammable substances, and they are also gases at room temperature. And you can see that they are simply made up of carbon and hydrogen in various formations there. And the reason that they are so flammable, they're storing a lot of energy in their bonds because of these very stable solid bonds that they have. So that is an important property. So here we have a question. Which of the following hydrocarbons has a double bond in its carbon skeleton? So the way that you would look at this question is to think about the fact that carbon is always going to fill its empty valences with a hydrogen if it has an empty valence position. So each carbon is going to, first of all, if we have more than one carbon, it's going to bond with an adjacent carbon, and then any leftover valences will be filled with hydrogens. So you can simply draw out, for example, the first one, 3 carbons in a row, and then actually fill all the empty valences with hydrogens. So I'll do that here and show you the correct answer. So the way to evaluate the answer to this is to actually draw these out. So I've drawn out a few of them here to show you. So A, if we have 3 carbons, first of all, they're going to make a carbon backbone, which means they're going to bond to each other. And then we have 8 hydrogens to place around. So you can look at this and say, okay, so carbon has 4 valences, so it wants to make 4 bonds. So this one, first carbon here, has a bond to the carbon next to it. Then it has 3 more available bonds, and so it adds 3 hydrogens. And now this carbon in the middle has 2 carbons it's attached to, so it has now 2 hydrogen spots to fill. And then this last carbon is like the first. It has only 1 bond to a carbon, so it still has 3 valences left, and so you fill those with hydrogens. And if you count up your hydrogens here, you see you have a total of 8 and a total of 3 carbons. So in this case, we all of these are being filled with single bonds, and we have a stable, good molecule formed here. If we were to draw out B and we had 2 carbons attached to each other and just 1 hydrogen available, that actually would not work because there'd be too many empty spaces or valence spots for both of those carbons to be filled. And so this molecule would need a lot more hydrogens or double bonds to fill up those carbons. So now if we move on to C, we have CH4. And so that means we have 1 carbon with its 4 valences filled with hydrogens. So this again is a good stable molecule. It has all 4 valences filled with hydrogens here. The C2H4, if we draw that here, we have 2 carbons. Again, they're going to bond with each other. And then we have 4 hydrogens. So we can put 2 onto each carbon. But if we count how many bonds that are around the carbon, we only have 1, 2, 3, and for this carbon, 1, 2, 3. So it's still, these carbons still have 1 empty valence. And so the way that's going to be formed in this particular molecule is to put a double bond between the 2 carbons. And by doing that, you've now made a stable molecule, and the carbons, each of the carbons has 4 bonds filling those 4 valences. Okay, so that's how you again would figure out this type of problem. So this is actually going back to Chapter 4.3 and the discussion of functional groups. And you really do need to memorize these and know them. So there are only 6 that you need to learn, and these 6 major ones are important. And just know these basic characteristics about them. So a hydroxyl is the OH. It is responsible for making the class of molecules called alcohols. Typically, a molecule is going to end with an OL, like ethanol, when it has these hydroxyls on it. A carbonyl is a carbon that's double bonded to oxygen. It can take the form like this one here, where carbon has a bond to something else over here, and then 1, 2 bonds to oxygen. So now we have 1, 2, 3, and then the 4th bond is to a hydrogen. So, but the fact that it has this C double bonded to an oxygen makes it a carbonyl, and that's the only portion that we're actually focused on. When the carbonyls are at the end of a molecule, like in this situation, it's called an aldehyde. If it occurs within the middle of the molecule, then that molecule becomes a ketone. Okay, the carboxyl group is the COOH, and it would form like this. Again, we have this carbonyl idea, but we still classify it as a carboxyl if what is attached here is another oxygen, or actually a hydroxyl attached. So we have the carbonyl with a hydroxyl, that makes it a carboxyl. Oh, and it's important to know that carboxyls, if you see that attached to a molecule, then that molecule is going to be an acid. Okay, the next 3. We have a sulfhydryl, which has the SH, a sulfur bonded to hydrogen, and compounds with this sulfhydryl are thiols. These are stable groups, and particularly when they're on proteins. They stabilize the structure of a protein. Aminos, speaking of proteins, are the NH2s that are attached. You find them on amino acids. Always. They're a portion on every single amino acid, this NH2. And this particular functional group is important because it's very good at accepting hydrogens. So it can act as a way to, almost as a base in terms of attracting excess hydrogens in solution and making this an NH3. So the phosphate is our last group here, and that's the PO4. The PO4, again, is a very important molecule in biochemistry. Because of these 4 oxygens around the phosphate, we have a lot of electronegativity. So this is a highly reactive functional group. It's a portion of ATP, which is adenosine triphosphate. So it has actually 3 of these phosphate groups. GTP is another energy storage molecule, which again has phosphate in it. And as you know, we have phosphates in nucleic acids. So they are actually, and they're also found in phospholipids. So we see a lot of this functional group in biomolecules. So looking at this particular molecule that you're seeing here, what functional group can you not find of the 4 listed? So take a second, pause this, and make sure you can identify the different functional groups here. So hopefully you were able to see that there is a carboxyl here. We have the COOH. We have a hydroxyl. We have an OH right here. We have an amino group right over here. But what we do not have is any sulfhydryl. So there's no sulfur molecules on here or elements on here. Okay, so next we're going to go back to Chapter 5 and start to break down the 4 biomolecules. So we have carbohydrates, lipids, proteins, and nucleic acids. And these are the 4 major classes of biological molecules. And all of these molecules, we're going to actually say that only 3 of these molecules can take the form of a polymer. But they're all considered macromolecules. And we're going to distinguish one particular group that actually does not make a polymer. But most biological molecules are assemblies of different monomers, single molecules that are identical. And these are formed by linking together these building blocks into long chains. And so you can see here some examples. A monosaccharide is a single molecule of a sugar or carbohydrate. And when we polymerize that, we add more than one of these together, we get a polymer, and we call it a polysaccharide. The next group is the amino acids, or the protein group. And an amino acid is the monomer of a protein. And when we string together or we polymerize these amino acids, we get a polypeptide or a protein. And a distinction that comes up a lot, or a question that comes up, is what is the difference between a polypeptide and a protein? And really, there is a subtle difference. They are essentially the same thing.